Welcome to SARC Talk. SARC is a nonprofit pan-sarcoma research organization that develops and manages clinical trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review innovative scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode number 2. My name is Scott Okuno, medical oncologist at Mayo Clinic and Chief Medical Officer for SARC. Our episodes are posted on our website, YouTube, SARC channel, and wherever podcasts are available. Today for SARC Talk, episode number 2, we're excited to have Dr. Inga-Marie Schaefer, Assistant Professor of Pathology, Harvard Medical School, and Associate Pathology at Brigham and Women's Hospital, and Associate Member of the Broad Institute. She's a member of the board of CTOS, the Connective Tissue Oncology Society. She is the 2022 Liddy Shriver Early Research Award. This is an award based on track record of early achievement for elite international nominated individuals. She is the 2019 SARC CDA Award recipient for her efforts of targeting cell cycle dysregulation in GIST. Congratulations on your success and welcome to SARC Talk, Dr. Schaefer.
Thank you very much, and thank you for having me.
Dr. Schaefer, your career path is a bit different than most. Can you share your career path and how you presently landed in Boston?
Yeah, that's right. So I'm originally from Germany. That's where I grew up, in a small town in the north of Germany, and that's where I went to medical school and also started working as a pathologist. So I underwent training in anatomic pathology at the University Medical Center in Göttingen. And my initial dream was to just be a surgical pathologist. And then things changed a little bit. It was actually early on in my career, even as a medical student and then as a trainee, that I got exposed to GIST. We had one of the largest cohorts of GIST in the country that was clinically annotated. So that really sparked my fascination for these tumors really early on. And then I decided to pursue a postdoctoral fellowship with Jonathan Fletcher at Brigham and Women's Hospital in Boston. And that's where I came in 2013, thinking that I was going to just do the postdoc for 2 years and then return to Germany to finish my training. But when I came to the lab, it didn't take very long that I wanted to change career paths. And I decided that I would love to pursue a career as a physician scientist in the US. So I finished my postdoc in 2015 and then redid my anatomic pathology training at Brigham and Women's Hospital and finished that in 2018 and then joined faculty in this department. And in summer of this year, I transitioned to becoming a PI and starting my own translational sarcoma research program in the Department of Pathology at the Brigham.
That is definitely a different career path, and clearly there was some things that have changed during your planning part. But right now you have a Schaefer lab, as you said, and you're actively recruiting, which is great. And you can give a plug for people that might be interested. But as you changed from your pathology realm and developing a lab, what were the surprises that you experienced starting your own lab?
Yeah, so there were a couple of things. So I think initially when you work as a postdoc in someone else's lab, you have much less responsibility and you can fully focus on the science, move the science along, get papers published. But when you suddenly start up as a new PI, there's suddenly many more responsibilities. So one of the things that I started to learn, and I'm still learning, is how to hire people for the lab, how to manage people, and how to best develop the people who work with you, because I want to be a good mentor and role model for them as well. Then you have to secure funding, so you have to be very prolific, write a lot of grant applications, and see that you are able to sustain your lab in the future. Then, of course, you have the science, so you have many new ideas and you have the great opportunity to really move these along in a very independent fashion. You can, which is actually what I love about being a new PI, you have all these creative ideas in your head and you are now able to really pursue those in a way that, you love. So that's the science part. And then I think there's also, leadership skills that now I have the ability to apply and also further develop, and also in terms of professional development. So there's all these aspects of the career that now come together, hopefully in a very synergistic way. Yeah, it's a great opportunity, and it's a very exciting phase in my career, and I really appreciate the opportunity to be able to have that lab, here at Brigham at Women's Hospital.
I can just tell from the excitement that you enjoy the challenges and the opportunities.
Yeah.
And we did say that your lab is actually actively recruiting, and we'll make sure post a link to your website that does allow for hire the interested in hires. One of the things that does come up, and you're learning, as you said, but there are a lot of young sarcoma clinical researchers that are starting to decide a career path that says, hey, should I do work in basic science versus clinical trials? And I know you say that something flipped in your mind is that, hey, I want to be a scientist and do basic lab. But we want people to hear a little bit is what is the most challenging aspect as you made that transition from a clinical pathology person to say, no, I really want to now run a lab, be collaborative, and what are those challenges that you see now?
Right. So I think as a pathologist, the clinical work is a bit different than for most clinicians, but still we see patients and patient samples, and in that sense, this work is pretty similar to other clinical specialties. But when I decided to pursue a career as physician scientist and not only as physician, it really opened up so many new avenues for me, which I think are challenging, but at the same time are exactly what I was looking for. So your day-to-day work, I think, is much more flexible and also in a way more unpredictable. So, when you work as a clinician or pathologist clinician, you have, a defined work schedule. You know exactly which patients you're going to see, which samples you're going to evaluate in a given day, and everything is very predictable in terms of your work schedule and the expectations for you. When you start a lab, I think you have much more independence to shape how you work. So in terms of work hours, in terms of how you spend your time and your efforts, on which projects you want to work and how fast you move them along. So really to have that independence, and I think it's challenging because it's unpredictable eventually how things go, but it's also, I think, the most exciting and rewarding part of being a scientist. So yeah, what I love is that you can follow your own ideas and be really creative in the lab, and take ownership of the work you do. And that's what I think is a big difference in terms of the kind of attitude to your work. When you compare yourself with being a clinician, where everything is much more predictable and foreseeable, and there's also much more routine work. I would say in the lab, you have to deal with many, setbacks, but, once you appreciate those, not as failures, but, as challenges for growth, it makes it much more enjoyable, I would say, to work as a scientist.
Yeah, it almost makes me want to become a scientist now to have autonomy and structure my day a little
Yeah.
bit more. But I think with that freedom, as you said, does come responsibilities, and those are the challenges. And as young people are coming through the system and understanding it, they do need support. And I know that in 2019, when you essentially had a developmental award through SARC. That's the first time I heard about you because when you go to the meetings, you see things that way. And
Mhm.
can you describe how the impact of that SARC CDA impacted your career?
Yeah, so I would say that the Career Development Award from SARC was really instrumental in my career at the time. So I was just, about to think about pursuing a career as physician scientist, and it was really crucial to support my research at the time, to give me the protected time that I needed to drive my research along. And it enabled me to not only generate a lot of research insights in Johnson Fletcher's lab, but also to apply for additional funding. So around the same time, I got a KO8 award from the NIH, which I'm still on, and it led to other, subsequent grant applications that kind of all build upon that work that was done under the SARC award. So in that sense, I think it was really instrumental in my early career, and I really appreciate the support that I got from SARC. And it also connected me with the people from SARC. So it was really great to be able to foster collaborations and network with other sarcoma scientists and clinicians in that way, and build, long-term relationships.
No, that is true. Getting connected in the sarcoma community and understanding who the people are and allowing the stepstones to the next grants that go
Right.
along. As we're talking to more and more awardees, we're finding that there is a huge amount of benefit, and the return on investment is great for the sarcoma community and SARC. Our goal is to, award more people that can have this, because this is the important aspect. Looking at your lab, the Schaefer lab, you're centered on biology, genetics, molecular mechanisms of sarcoma to develop novel therapeutics. Uh, you say that you're combining genomic, structure, functional, and multiplex imaging studies to uncover biological mechanisms, including cell cycle perturbations. That's a lot. And, looking at the literature that you've published and the papers that you've published, you're starting to characterize a genomic progression specifically for GIST. And you describe an 8-step process that you have
Right.
developed so far. You've published on the 14q max in GIST. Can you follow up a little bit more on how all you work through all those things and just progression?
Yeah, so that work was kind of started when I came to Johnson Fletcher's lab as a postdoc. So that was a main focus of my work at the time, to really understand and decipher the steps of progression that just undergo as they develop from early lesions and low-grade tumors to eventually high-grade and aggressive and metastatic cancers. And we and also other groups have observed that just acquire losses mostly on chromosome arms as they progress. So we were then trying to understand in which order these events occur during progression, and you could kind of establish a sense of that when you look at the frequency of these aberrations in low risk, intermediate, and high risk GIST. So we identified these 8 steps that kind of build upon each other. The initiating step is the tyrosine kinase mutation. Then, as you mentioned, the 14q loss that inactivates the MAX tumor suppressor, which is followed by 22q loss that inactivates DAPDC5 and mTOR regulator. Next, we have losses usually occurring at chromosome 1p, and we still don't know what the tumor suppressor is that is, the putative 1p tumor suppressor, because this chromosome arm is really long and it's a bit challenging to identify that candidate, but that hopefully will be identified at some point. And then we have next a cell cycle regulator mutations, losses at 15q, where we are also currently, unclear of what that tumor suppressor is. Then we have inactivation of dystrophin, and then finally the secondary tyrosine kinase mutation occurring in the metastatic setting. So we identified MAX as the 14q tumor suppressor and the second event in GIST progression. And MAX inactivation, as I showed in my postdoctoral work and in the paper that we published at the time, leads to downregulation of P16 and early cell cycle dysregulation. And as I just mentioned, high-risk GIST also develop then mutations that directly inactivate key cell cycle regulators, such as CDKN2A and B. And we got really interested when we observed that out of these 8 events that I just mentioned, 3 repeatedly target the P16-RB1 pathway. So this really told us that this pathway must be key in GIST development and progression. I think overall this is a beautiful concept, and it shows that GIST are I wouldn't say unique, but quite special amongst the sarcomas, because most sarcomas develop with one fell swoop. So they have one translocation that then initiates the sarcoma, and it's there. And we cannot really identify steps of progression, but I think GIST are a bit unique in a sense that they develop from the Cajal cell lineage. So we have the originating cell type, and we know that Cajal cell hyperplasia and microgysts are precursor lesions to clinical GIST. So we have this whole range of progression in GIST identified, which I think is a beautiful model to work with.
No, you're right. And delineating that nice progression that you talk about, the steps that are needed to eventually get for growth and metastasis is
Right.
key. And, when a lot of us were in training, that was the same philosophy in polyp formation for colon cancer, and people have worked that out. Can you describe how this might impact other sarcoma types and the rigor that you went through with GIST, or is this just unique for GIST?
I think it's not unique for GIST. So there's another sarcoma type, like malignant peripheral nerve sheath tumor, MPNST, that also shows a comparable progression like GIST. So these tumors originate from neurofibromas that then develop into atypical neurofibromas, and then low grade, intermediate, and high grade MPNST. And in parallel to GIST, there have also been certain steps of progression identified in nerve sheath tumors. So I think in that sense, this model of progression can be translated to certain sarcoma types.
No, that is a good model, and I think there could be opportunities in the 100 other subtypes that we have for
Right.
sarcoma to start working through that. And from us as a clinician, we're always looking for new drugs, our new mechanism target. And I know that you've published work recently in the role of CDK2 and CDK4/6, and maybe can you describe the story of how you came about that and the implications as we go forward in the treatment of gastrointestinal stromal tumors?
Yeah, so what we just talked about, this model of stepwise genomic progression and the identification of MAX as an early tumor suppressor in GIST, really naturally led to the subsequent discoveries from our group. So we early on recognized that cell cycle dysregulation that occurs in a stepwise fashion seems to be very important in GIST. And when we further looked at the cell cycle pathways, one of them being mostly regulated through P16, CDK4/6, and then RB, and the other one signaling through the P53 CDK2 pathway towards RB1, it really became obvious that the mutations that GIST acquire during progression really target both of these pathways. So oftentimes GIST don't only have P16 inactivation, but also concurrently an inactivation that targets this P14, P53 pathway. And this was really striking to us. And in fact, we then analyzed multiple GIST clinical samples and also cell lines, and found that mutations that dysregulate the CDK2 and CDK4/6 pathway concurrently are present in virtually all high-risk and metastatic GIST. So this showed us that this is important. And there had been a study by the French Sarcoma group, I think in 2019, where they tested the CDK4/6 inhibitor palbociclib as a single agent in advanced GIST. And they found that as a single agent, this inhibitor was not effective. So our hypothesis was that, as in breast cancer, where you combine CDK4/6 inhibitors with other biologically rational drugs, you would ideally also want to combine them with a second drug in GIST to maximize response. And since we had seen that the CDK2 pathway is often co-dysregulated with the CDK4/6 pathway, our hypothesis was that combination of CDK2 and CDK4/6 inhibition would have maximized effects in GIST. And indeed, we tested multiple inhibitors of CDK2 and CDK4/6 alone and in combination, and also a combined CDK2/4/6 inhibitor, and found that combined inhibition indeed has synergistic effects. It inhibits RB1 phosphorylation, so that's really important because dysregulation of CDK2 and CDK4/6 in these tumors results in inhibition of RB1 tumor suppressor function. So the goal of any therapy is to kind of restore that tumor suppressor function and slow down proliferation. And we also found that combined inhibition had synergistic effects on cell proliferation as well. So yeah, this was a very nice finding, I would say, which we are currently confirming in vivo as well.
Yeah, you bring up the point that with the base that you had with the progression, it was a logical target for CDK4/6. And then you pivoted with a negative study done elsewhere to figure out why wasn't it effective, and then you're starting to work through the
Right.
CDK2 mechanism, and this is how the field moves along. And it's nice to have that basic science component that can
Right.
rationally figure this out, and the clinical aspect too. So it's a neat story. So we look forward to in vivo and then obviously clinical trials on
Yeah.
this as well for
Right.
our patients. So I know we always want to be in a rush to get it to trial, but there's a good amount of science in there. So good.
Yeah.
The other thing that your group is working on is tumor suppression in leiomyosarcoma, and part of SARC is collaboration. And I know that in last year you did publish your work in highly recurrent tumor suppressor alterations in leiomyosarcoma. You did work collaboratively with MD Anderson. That also contributed with the patient advocacy groups to Stanford, and you did get support from the SARC Leiomyosarcoma Research Fund, the Liddy Shriver, the Leiomyosupport and Direct Research, as well as the National Leiomyosarcoma Foundation. It is a big effort, and the collaboration was key. And um, part of what we do at SARC is do patient advocacy. And I know that the two main groups that were helping in this part were helpful. Maybe you can describe a little about how how those patient advocacy groups help support the paper and the samples.
Yeah, so that was a work that we published in 2021. And as you said, it was a collaboration with MD Anderson and also Stanford University. And the samples at Stanford University had been contributed by patient advocacy networks. And we were, of course, very grateful for their contributions because without these, this huge study would not have been possible. So we were able to collect I believe, more than 500 LMS samples. And in the final study, we included 489. And that was to date the largest sample size for a study in leiomyosarcoma. So we were really grateful to the support we received from the patient and patient advocacy networks that enabled that study. I think in the rare sarcoma field, in the rare disease field, sarcomas are rare diseases, I think it's really important to collaborate, not only between clinicians, scientists, and diagnosticians, but also the patients who are affected by the disease and who really make a tremendous effort, oftentimes, to organize themselves, educate the community, and support research in the field. So I think it was a great example of what collaboration between all these stakeholders can achieve when we work together effectively. And I think it's really important for us physicians and scientists to continue to work closely with patients and their advocacy groups because we can learn a lot from them. And it's important to not only, include them on a paper or a grant application, but really have that continuous exchange of ideas and so that we can both learn from each other what are the expectations and what are the special needs that patients may have that we may not be aware of. So I think it's really great to, have that collaboration be ongoing, and involve each other whenever possible to have the best, impact on the field.
We're very fortunate to have very active members in GIST as well as leiomyosarcoma from patient advocacy. And, we would hope that all tumor groups had active groups. And, it does, like we're saying, is collaboration historically has just been with physician and institutions. but I think we missed out a lot, and having them engage allow 500 samples. My gosh, that is the
Right.
largest study that was done, and it's a great paper, and it kind of starts setting some framework to go along. And without the help of patient advocacy, it would have been less impactful in the number. So
Right.
we do
Hmm.
appreciate that. The other aspect is, you just don't work in Boston. You work at other major sarcoma centers and collaborations that you do have. And what advice do you give to young researchers that are trying to set up these collaborations?
Yeah, so my advice would be to be open, share your own research and ideas with others who might be interested, go to meetings, take the opportunity to present at conferences and really, share your work with others. Um, and yeah, use any opportunity you have to network, get to know other scientists in the field, use the opportunity to learn from them. How do they set up their research program? Yeah, and when you get in contact with others, you learn about synergies that might evolve from your research working together with others. So yeah, be open-minded, try to learn from the important players in the field, get to meet new people as you go along. And I think when it comes to picking collaborators, it's important to pick people who you like and who you like to work with. So I think it's important to get along because you will spend a lot of time together. And if, you know, sarcoma science is the work you love to do, which I'm sure is the case if you're passionate about science, you want to spend that time with people who you like to work with. So I think, the personal aspect is also very important. Not just the qualifications, but also, if you find nice people to work with the nice people.
Inga, I didn't even think about that, and it's probably important for us as collaborators to work with people you like because you'll get
Yeah.
more out of that. In addition, this is a long-term relationship that you want to develop. So that is very insightful. And it's probably more important than anything is to develop a friendship and a collaboration.
Right. And I think it's important, to have people who you trust. So a trustful relationship, I think, is key for any fruitful long-term collaboration.
I'd be remiss if we didn't dive just a little bit into the paper that what was done,
Yeah.
and maybe you can summarize your findings of the primary versus non-primary LMS alterations and how we should approach primary disease different than metastatic disease based on your paper.
Right. So we primarily looked at expression alterations of the following markers. So we analyzed P53, RB1, P16, and P10 aberrations. And in the large cohort of more than 400 cases, we included both primary and non-primary LMS, and also uterine and soft tissue LMS. And what we found was that virtually all non-primary LMS have aberrations of P53 and the P16 RB1 pathway. And when we compared primary and non-primary LMS, it was actually interesting to see that there was no significant difference between these aberrations. So it seems that alterations of P53 and P16 RB1 are already very frequent in the primary LMS. So that told us that, first of all, these are likely early and non-evolving events in LMS development that are not only present in the metastatic setting, but already in the primary tumors. And this also led us to conclude that these markers can be used as biomarkers not only for metastases or recurrences, but already when you have the primary LMS specimen available. So in that sense, I think they should not be treated differently, and you can actually use the findings in the primary LMS as a surrogate what's really going on in the tumor, and you don't need to wait until you have, recurrent or metastatic disease to analyze that sample for any important biomarkers.
Yeah, that's an important aspect as all of us are trying to do biomarker driven studies.
Right.
The mechanisms that you looked at are there in the primary as well as the metastatic disease. Clearly, more work needs to be done because there's an evolution. We just haven't figured out what are those additional changes that allow it to go from a
Right.
primary to metastatic. But this is the largest paper really the foundational aspect that will help impact us as we develop clinical trials and or biomarker-driven trials. So this is very, very helpful. There are many groups looking at clinical trials, and from your work in GIST, your work in this LMS space, do you have any suggestions for like where do you think the next planned study should be if you're thinking about let's focus on leiomyosarcoma?
Yeah, so since we found in that study that virtually all non-primary LMS have aberrations of the P53 and also P16 and RB1 dysregulation, we feel that therapies that target either cell cycle aberrations and or DNA damage repair vulnerabilities should be further explored in LMS. And I know that some groups, including Jonathan Fletcher's group, are currently working on that, trying to initiate clinical trials that really leverage the DNA damage repair deficiency in LMS. And also these tumors are characterized by very complex karyotypes, so they are genetically very unstable, and this can be leveraged. But this is currently tested in clinical trials. And yeah, hopefully there will be some important leads coming out from these studies in the near future.
Absolutely. And once again, knowing the biology, knowing the mechanisms allow us to figure out what the next important trials are going to be.
Right.
Dr. Schaefer, you are doing a lot of work here. You did get this 2022 Liddy Shriver Award, and all of us were excited when we heard about it. We saw you give your presentation at
Thank you.
CTOS. Uh, in it, I'd like to hear a little bit more about that aspect, but you're also part of a developmental therapeutics group as well. And I'm just trying to think about how do you find time to do all this?
Yeah. I think if you love your work and if you're passionate for what you do, it doesn't really feel like work. So yeah, I enjoy working in the sarcoma field and all different aspects of it, be it in the lab, be it collaborating with other physicians, clinicians, and scientists. So yeah, I don't know. I somehow find the time, and there's a lot of overlap actually. So this is a developmental therapeutics program that's part of the Dana-Farber Harvard Cancer Center here in Boston, and it's kind of a multi-institutional program where members from the Brigham and the Dana-Farber can participate in. They have different programs, and I'm part of the developmental therapeutics group that includes many members that are oftentimes also physician scientists. So everyone brings a different perspective to the field. And I think it's also great because I, for instance, work on sarcomas, but there are many other clinicians, of course, who work on breast cancer or colon cancer and other disease types. And it's a great opportunity to learn from each other how they approach the most important questions in the field. How do they generate the basic science insights that we need to then identify vulnerabilities for treatment and then leverage this to hopefully develop new therapeutic avenues. So yeah, I think it's a great program, and there's a lot of resources to collaborate and network with each other and share resources and core facilities as well.
Yeah, I mean, it's almost like everybody wants to be at your institution because of we can get into a silo only doing sarcoma. And as you said, cross-pollination does
Mm-hmm.
allow for broadening your horizons, but it takes time and effort. And clearly, you're learning a lot, contributing a lot to that group as well. And it did, at least from the sarcoma community, the Liddy Shriver Award was quite impressive. And just some thoughts on that from your standpoint.
Yeah, I am still very honored to have received this award this year. It comes at a very important time in my career where I'm just starting off my lab, and I feel very honored and grateful mostly to Bruce and Beverly Shriver for their recognition of my work and for their support of my research over the next few months and years. So I'm really grateful for this recognition. And I think I think it's also great that the awardee is usually someone who has a track record of collaboration in the sarcoma field. And as I said, I think that's really important whether you're a pathologist or an oncologist, or surgeon, or radiation oncologist, basic scientist. I think it's important to really appreciate that we all have a common goal, which is to make a difference for sarcoma patients and really work together as good as we can to, educate each other and make a difference and move the science along. So in that sense, I really appreciate that award, and I look forward to, expanding my network and expanding my collaborations with other sarcoma clinicians and scientists in the field.
No, we do appreciate it. Well deserved. And we're looking forward to many more things coming from you and your lab and your collaborators. Dr. Schaefer, are there any additional thoughts or comments that you have at this time?
I think I would like to thank you for inviting me to this podcast, and for SARC for giving me the opportunity to meet with you today and exchange our thoughts in sarcoma. And I look forward to updating everyone over the next months and years on how things are going in the lab. And I look forward also to foster more collaborations with everyone in the field. Thank you.
Well, Dr. Inga-Marie Schaefer, it's been a pleasure learning more about your career path, your lab, your thoughts and work in leiomyosarcoma and sarcoma in general. And again, we'll look forward to more and more collaborations as we move forward.
Thank you so much.
Thank you. We look forward to our third podcast that's going to be coming up. So look forward to those announcements as we go along. Until then, thank you all, and we'll see you again after the holidays.
Thank you for listening. Be sure to subscribe so you never miss a SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or ask questions, please email us at sarctalk@sarctrials.org. And remember, together we can find a cure for all forms of sarcoma.